Our next speaker is Professor Tess Lambe. Tess is Professor of Vaccinology and Immunology at the Jenner Institute at the University of Oxford and is one of the co-developers of the Oxford AstraZeneca COVID-19 vaccine. Importantly, the Lambe Group are also currently developing and testing vaccines against a number of other potential outbreak pathogens, including Crimean Congo hemorrhagic fever virus, Lassa virus, Nipah, influenza, Ebola virus, and coronaviruses in general. A number of these vaccines have progressed to clinical trials and may be important for future emerging outbreaks. And with that, Tess, I wonder if you might like to share your slides. Thank you very much.
I shall try. Can you see the slides and hear me well?
Perfect.
Great, thank you, Emma. First of all, thank you to the organisers for inviting me here to speak. I'm going to talk about the lessons that I've learned from COVID-19 and the journey that I've had across the last 2 or 3 years. So a little bit of a disclaimer that my journey will be different to everybody else who's worked in the clinical trials. I want to take you back to the Ebola outbreak that started in 2013, 2014, and went on to claim an estimated 11,000 lives, an estimate that is largely thought to be underrepresentative of what actually happened. And after the Ebola outbreak in Western Africa, there was a number of task forces set up. The WHO and other government bodies generated a hit list, for want of a better word, of those pathogens that they thought might generate the next pandemic, epidemic, or outbreak. And there was a call to arms. There was a call put out to generate vaccines and therapeutics against all of these pathogens of concern. And some of these, I suspect, a large number of you will have heard of. Everybody's probably heard of Ebola virus and Zika, Crimean Congo hemorrhagic fever, which I think is a favorite virus of both myself and Emma's. Doesn't tend to roll off the tongue, but is very impactful. And Rift Valley fever as well is a little known virus. Regardless, we at the University of Oxford and many other places, including some pharma companies, started to make vaccines against these pathogens of concern. So I wanted to go into a little bit of detail, not too much, about how to make a vaccine, because it is relatively easy. It's not the witchcraft that some people seem to think it is. But first and foremost, you need to know a little bit about the pathogen that you're trying to make a vaccine against. And that's where genomic surveillance and sequencing is hugely important, so we can identify the pathogen that is causing the outbreak. And once you've identified the pathogen of interest, what you really want to do is identify a part of that pathogen that you think, if you have a strong immune response against it, you'll stop infection. So Sharon has already covered some of the details of the spike protein, and that was the most obvious target for the vaccine manufacturers to use in making a vaccine, because the spike protein is how the virus gets into your body. And if you can neutralize that threat, then you're going to be able to protect your vaccinees, your volunteers in your clinical trials. And two platform technologies, vaccine platform technologies that really came to the fore during SARS-CoV-2 pandemic were viral vector vaccines and RNA technologies. And in essence, they both work in a similar manner. They both take the recipe or the coding sequence, if you will, for the spike protein, and they get it into your body. And once these platform technologies has carried the recipe for the spike protein into your body starts to make large amounts of spike protein. Your immune system recognizes it as foreign or alien and generates both antibodies and T cells, and hopefully to a level that you will be protected when you see the actual pathogen, when you come across SARS-CoV-2. And really, these platform technologies were seen as the darlings of the COVID pandemic. They were the ones that were hailed as revolutionary, as the next best thing. But I really think one of the lessons we need to take away from the pandemic is not to de-invest in the other technologies. Whole inactivated vaccines and protein subunit vaccines have a huge part to play in preventing infections routinely and are used routinely in our healthcare system within the UK. So while viral vector vaccines and RNA technology have got a lot of attention across the last 2 or 3 years, one lesson that I have learned is that we can't de-invest in all of these techs because we don't know what is coming next and we don't know which technology may work better. And truth be told, viral vectors have been around and RNA for decades. In fact, the first viral vectors that were licensed were vaccines that were licensed against Ebola virus. And they were trialed and tested in the outbreak that happened in 2013 to 2016. A question that we frequently get when we're giving talks is, how were we able to go so quickly when it normally takes you 5 to 10 years to generate a therapeutic or a vaccine? And to speak to that, I thought I would show you what the normal development process and timeline looked like before COVID. And this was a linear process where you started with designing your vaccine that would generally take weeks to months, depending on how complex your pathogen was. It can be sped up. Myself and the other co-designer designed the Oxford AstraZeneca over a weekend, so you can do faster. The next step is to start doing preclinical studies, and you normally start with small animal studies here, and you're looking to see if the immune response is such that it is the type of immune response that you would want. So typically neutralizing antibodies and a good T-cell response. And if you don't get that response, then you go back to the drawing board, you start designing your vaccine again. Because really, as you progress along this timeline, each of these steps are not a small increase in price or time or investment, they're astronomical. So we're quite lucky in Oxford in that we have a we have a GMP manufacturing site where we can make vaccines that can go into clinical trial. But even so, those vaccines will generally cost between $1 million to 1.5 to generate. And by the time you've got to phase 3 GMP or onward licensure, you really want to make sure that you've got the best in class product, that you're progressing what you think is a game changer, a market changer. And to do so, you need to invest a lot of time, a lot of effort, and a lot of money. And that is typically why vaccines or therapeutics take 5 to 10 years to develop. We looked at this at the start of the COVID pandemic, and we realized we couldn't go that slowly. So how did we actually go more quickly? Well, we took a risk, and the risk we took was purely financial. And it wasn't a risk that I took, it was the UK government's money, it was Serum Institute, AstraZeneca, and other investors that allowed us to start our phase 2 and 3 clinical trials while we were still running our phase 1 clinical trials. And I hasten to add that while I say we did work at risk, we always prioritize the safety of our volunteers. I suspect speaking to this audience that that goes without saying, but it doesn't help reinforcing that point because we frequently get asked that question. So another lesson that I've taken away from the pandemic is if you're going to go quickly, you need some plans in place, and you need to plan for the future, and you need to plan many steps ahead because things will, can, and do go wrong. You absolutely, to my mind, need to partner with pharma early, and you need collaboration. Without that open collaboration between government regs and international bodies, there is no way that there could have been a vaccine in less than a year. So those silos, those walls came down between all of those key stakeholders, and we were all pulling in the same direction. So to my mind, that was one of the most important outcomes, is collaboration and open dialogue that allowed us to make a vaccine in less than a year. So what were the other types of learnings that I've taken away from running these clinical trials? Well, to set the scene, we made our first batch of GMP, our first doses were made and filled in early April. The vaccine was released to trial towards the end of April. And at that point, we had a number of very lovely volunteers who even came to clinic without sleeves, so they didn't have to roll them up, ready and able to get vaccinated in their arms. We partnered with pharma, again, relatively early on the 30th of April. And what had started to happen across those early months really took off when we started our clinical trials. And that was the fake news and misrepresentation and downright lies in some cases around what was happening in these trials. So our lovely volunteers who came forward and were vaccinated woke up to fake news that they had ceased to exist 3 or 4 days after being vaccinated. And one of the tough lessons to learn was no matter how much you try and do to dissuade or get that genie back into the bottle, get that fake news disproven, it doesn't matter. Once it's out there in the ether, there will be people who believe it, there will be people who will propagate it. So we really need to tackle this head on, and we probably need to do it in a more proactive way than we necessarily did during the pandemic. I have to admit that it was a steep learning curve for me and many of the other team members in communication and how to get out front and in front of these stories. The other point that I would like to make is around the efficacy readouts that came towards the end of 2020. And sometimes science is difficult, science is hard, it's not easy to explain, and certainly our efficacy readouts were not easy to explain. But if we don't do it well enough the first time, then we should go again and we should explain it so that people can understand. And I'll take this opportunity to run you through some of the confusion around why we had 3 numbers and everybody else had come out with just 1 number. So within the UK, we had 2 different vaccination regimes. We had individuals who had had a low dose and then a standard dose. And we also had individuals within the UK and in Brazil who had received 2 standard doses of our vaccine 4 weeks apart. And then efficacy readout that was associated with those individuals that had 2 standard doses was in and around 60 to 64%. And for the low dose, followed by the standard dose, it was much higher at 90%. At the time, we had done this analysis over a weekend, and it had been independently verified. And when you are working with a large pharmaceutical, there is an onus on you to come out with these results as soon as you know them, because they can impact share price. We weren't hiding data. We didn't know why there was a difference between those regimes. We were able to demonstrate after a couple of weeks or some time that the reason there was a difference between the efficacy associated between those 2 regimes was largely down to the interval between the doses. So in those individuals who had had a low dose followed by a standard dose, there was a longer interval. And that longer interval was associated with a higher antibody titer and a higher efficacy readout. So really, again, communication was critical. And would I go back and try and simplify the message and give 1 number instead of 3? No. I think sometimes we need to be driven by the science, and science is hard, and we need to explain it. The one thing that I would suggest that I would ask the audience, dissuade them from doing, is comparing across clinical trials. Everybody does it, including myself, but it's wrong. Generally, clinical trials are set up for a snap period of time or in a certain group of people, a defined population with a defined variant, and also the primary outcomes and how we define disease versus other manufacturers are different. So really comparing across trials, you're not comparing like with like, and you're doing the data a disservice. We were really encouraged when these vaccines went out into the wild, and they did what they should have been doing, protecting our most elderly, our most frail, and making an impact against COVID in older adults and the most frail individuals. And this certainly was a lesson that I learned, looking to see real world data to back up your clinical trials. It was really impactful and something that I still remember to this day. The other thing that I've learned is that I need to become, as we all do, better communicators. The importance of communication was really highlighted around the level of misinformation that got out there. The anti-vax movement that really tried to dissuade individuals from having vaccines. And anti-vax is very different to vaccine hesitant and vaccine reluctant individuals. Vaccine hesitancy, in my experience, generally stems from people who have questions. They don't quite understand something, they're curious. Having an open and honest dialogue with those individuals can help them decide whether they will or won't have a vaccine. They're generally very reasonable individuals who just don't know what to do. The anti-vax movement is something different. It's a different beast. However, anti-vax sentiment is nothing new. This is a cartoon from the time of Edward Jenner. Edward Jenner is frequently referred to as the grandfather of vaccination, although there were many people doing variations of vaccination before and at the same time of Edward Jenner. Regardless, he noticed that milkmaids who had been exposed to cowpox did not go on to develop smallpox, a devastating disease claiming an estimated 300 million lives. He performed some what we would consider relatively crude clinical trials in the late 1700s and 1800s, and was able to demonstrate that if you were exposed to cowpox, you would be protected against smallpox. And this was a cartoon at that time suggesting that if you were to get Edward Jenner's vaccine, you would go on to develop bovine-like characteristics. Nobody has turned into a cow because of vaccination, but this type of sentiment, this type of message was not dissimilar to the type of messaging that was being propagated around the Oxford AstraZeneca vaccine and it being called a monkey vaccine. Having said all of this negative messaging, I do want to highlight that we received many, many words of thanks, many very generous gifts, one being very generous brownie delivery. So for every bad message we got, there were lovely members of the community and the public who did reach out to us to thank us and support us. But fundamentally, I don't know that we're doing a different, we're approaching the anti-vax movement in the right manner. I don't know that we're doing a better job at actually tackling misinformation and doing it in such a way that we can help individuals who are hesitant or reluctant make a decision in the best informed manner that they could be. The other message or the other take-home point that was very evident to me and many others during COVID was the slow rollout of vaccines to lower middle income and low income countries, and how when the vaccines started to be generated, that most, if not all of them, were going to upper middle income and high income countries. That needs to change for the next pandemic. Certainly the Oxford AstraZeneca vaccine, the ethos was to generate a vaccine for the world. This is old data, but something that the team are hugely proud of, especially that 2/3 of our doses went to low and lower middle income countries, and that in excess of 3 billion doses were released to supply. But we really do need to tackle this disparity in where the vaccines went better, because without us all being protected, no one will be protected. So to end in a similar note to Sharon, what were the lessons that I've taken away from concept delivery of these clinical trials? Well, when a rapid response is required, it absolutely necessitates creative approaches to funding and research. And for that to be properly enabled, we need collaboration across government regs, charity, industry, and academia, and we need an open and frank dialogue. We absolutely need local and international capacity, and we need that to be a sustainable model. We need international capacity to run trials, to manufacture vaccines, to roll out those vaccines, and a strong scientific base in those countries that were seen to be left behind during the SARS-CoV-2 pandemic, so that doesn't happen again. How do I think we'll achieve that? Much to what Sharon was saying, we need to invest in people and places. We need an open dialogue, we need sustainable funding, and we need in-country investment to really get the capacity built up and maintained. And in that way, I think we will be in the best position going forward for the next pandemic. It's only when it happens, not if. And in that way, we will be proactive and not reactive. And with that, I don't have a slide with everybody's names, unlike Sharon, because I will forget somebody. So I take the easy way out and just put a general icon up for everybody. And thank you very much for listening. I'm very happy to take your questions.